Once again, warm good afternoon to one and all. We will continuation with our previous class about the staining of microbes. So in earlier class, we had discussed about the staining of bacteria. So now in this class, we will discuss about the important method of staining of fungi, because fungi is also important and that requires something different procedure for staining. So in today's class, we will discuss about the important staining method that is generally used for staining the fungus. So how we will proceed in today's class, the objective of today's class, that is we will discuss in depth about lactophenol cotton blue. In abbreviation, it is known as LPCB staining, and this is generally used to stain the fungi. And in exam also sometime question may ask that which stain is commonly used to stain the fungi. So you should know that the stain is specific to stain the fungi, and there is one media that is known as dextrose agar, that is also specific for fungi cultivation. So these two things you have to remember when we are talking about fungi in basic, that which media is generally used to cultivate the fungi and which stain is used to stain the fungi. So in today's class, we will move in this way. Like first, I will give you some introduction and we will discuss in brief about the principle of this procedure. Then we will go about the procedure. I had also tried to keep one video to illustrate this procedure for lactophenol cotton blue staining. And at the end, we will see about the important fungi of veterinary field. So first, let me recall about the last various staining about the bacteria. I had discussed in that class that basically the bacterial stain are divided in three groups. One that is known as simple stain, second that is known as differential stain, and third that is known as special stain. I had also discussed in depth in my last class that what are the examples of simple stain, what are the example of differential stain and the special stain, and how they differ from each other. So this is just narration of all this stain. Those comes under simple stain, those comes under differential stain, and those comes under special stain along with some photograph. So we will not discuss again about this thing. Now we will move to the next one, that is the staining of fungi. To stain fungi, the principally we are using lactophenol cotton blue stain. So lactophenol cotton blue stain is generally used to stain the fungi, and why this specific stain is generally used, that we'll see in coming slide. So first, let me give you some introduction about this stain. Generally, this lactophenol cotton blue that is generally used to stain yeast, mold, or you can say generally all type of fungi. So we can stain even yeast as well as mold. And this particular stain contains three important components. The first component, that is lactic acid. If you can see the expansion of LPCB, lactophenol, cotton blue. So L, that stands for lactic acid. So that stain contain the lactic acid. That lactic acid will help to preserve the structure of fungi. So when we are processing, that will try to keep the minimum disturbance to the original structure of fungi we are studying. So that actual structure you can or actual structure and actual organization you can reveal, and based on that you will help them to classify that particular fungi or identification of that fungi. The second important component of this one, that is P, that is phenol. So this phenol added in this stain because that will kill the live organism, because fungi is considered, many times it is considered more dangerous than bacteria, because fungal spore, they can easily contaminate your environment, you can easily inhale them, and that may leads to zoonotic disease. So whenever you are handling fungi plates, you should take proper biohazard and biocontainment procedure, and it's better to handle the pathogenic fungi in biosafety cabinet. So once you are adding your fungi sample with phenol, phenol will kill the organism, and now that is safe to handle outside biosafety cabinet. And this is also advantage. We had seen that the advantage of fixing this slide in bacteriological staining. So here the phenol is act as a fixative agent, and that will kill the pathogenic fungi, and that will remain as not infectious to the handler. The third and important, that is a cotton blue. So cotton blue, that is a stain that is generally added in lactophenol cotton blue, that has principally bind to chitin, and chitin, that is a characteristic of fungi cell wall. So fungi cell wall contains the chitin, and the chitin is stained by the cotton blue, and that's why your fungi at the end you will observe has the blue color. So here again you have to recall that what cell wall contain what principal component. You have to recall about your gram staining, that gram positive bacteria contain peptidoglycan, gram negative contain your LPS. Then you have to discuss, you should recall about your acid fast staining. Cell wall contain mycolic acid like mycobacterium. So similarly here fungi cell wall contain the chitin, and that's why this cotton blue is selectively stain the fungi spore and fungi hyphae. Now moving this with this background, we will move toward the procedure of lactophenol cotton blue. So procedure of lactophenol cotton blue, that is generally it can be used as a wet mount or as a stain. As a stain means you have to prepare the smear and you have to go for staining. And wet mount means what I am going to discuss in next slide. That generally we are using wet mount. It is easy to stain. Principle and procedure is also very simple. Generally the important steps are like this, that once you beginning, you should have a clean glass slide and cover slip. Over this glass slide and cover slip, you have to put 1 or 2 drops of lactophenol cotton blue stain. With this stain, you have to add your fungus culture, mix it so that you will get the uniformity and the maximum fungi spore get separated so that it is easy to visualize, and then put a cover slip in such a way that there will be no bubble trap, and it will give a uniform thickness for easy observation. Now at the last we have to go for observation, and in general such slides were observed initially under 10x, and once you will see this spore, then you can move towards the 40x. You need not to go for oil immersion or 100x in this particular staining. In contrast to this, if you will see the bacteriological slide, bacterial staining, generally we are focusing at lower x, lower micro, lower that lens, and then we will move towards the higher or oil immersion lens. So this is one difference from bacteriological stain. Now sometimes there are some procedure where we can use the adhesive tape, by which we can put up or take up the fungi, and we can mix or we can put over the drop of lactophenol cotton blue, and over this adhesive tape you can put one more drop of lactophenol cotton blue and put up the cover slip, or you can directly go for observation without putting cover slip. So this is generally helpful when it is difficult to pick up your fungus from the plate, because they are penetrated inside the agar, and it will be difficult to pick up with your platinum loop. So in such fungi, it will be easy to take the samples by using adhesive tape. So if you will perform this simple staining, that is known as mount method of lactophenol cotton blue staining, then at the end you will observe fungi has a blue color and the background has a faint pale blue color. So that can be easily differentiate. That can easily differentiate. Okay. Now that how, here I had tried to put a one video where you can see that how we can perform this particular procedure. So it's self-explanatory. It has some caption that you can read, and in between I am also helping you to make it easy to understand. The first is the identification of fungi is mostly rely on the structure, and that require lactophenol cotton blue. So this stain generally we are using as a mounting stain. So first we require the stain, our culture, cover slip and slide, that you have to prepare ready, you have to keep it ready. Then you have to take one small drop of lactophenol blue with dropper over the clean glass slide. So this is sufficient to stain loop full of fungus culture. Now, after keeping this drop of lactophenol cotton blue, you have to take the fungi culture. To take this fungi culture, you have to sterilize your needle. Here, platinum loop may not help because they are very hard to take up from the agar. So, you have to sterilize your you have to sterilize that needle over the Bunsen burner, allow that to cool. Okay, once it will get cool, then you have to take the fungus culture from the edge of your fungus growth. Edge, because here there is a younger colonies and that can easily stain with your stain. Now with other needle, you have to scrape out all the culture what we are taking. After taking this one, you have to mix it over this lactophenol cotton blue. Once you will mix it, okay, so that everything gets separated, you will get the uniform thickness. Before this, you have to sterilize. You have to sterilize your this needle so that it can be reused. So now you have to put a cover slip over this one, your mixed one, in such a way that there is no air bubble get trapped. If you feel that there is a excess cotton blue stain, then you can take out some stain with a filter paper or by some other method. So this is a simple procedure by which you can stain your slide and you can go for observation under low power, then you can go for high power. Okay, so this is a procedure, by which we can observe the fungi. Okay, like this you can observe at the end of your staining procedure. So this is generally the video by which I tried to convince that how we can move, how we can perform a lactophenol cotton blue. Now moving to the next one, that what or how we can see, this one, that once you will observe under microscope, then how the important veterinary important fungi will looks, because generally look or based on their morphology we can classify. Okay, so before this I would like to acknowledge this website, because I took this video from this website. So I thankful to this website for giving me opportunity to share this procedure to my student. Okay, now coming to the next slide, that is about, okay, sorry, so next one, that we can see that what are the important one. Okay, so generally I kept the two. If you will observe, it will looks like this. This is a characteristic of mucor that cause disease in animals, and this is a characteristic of penicillin. So similar way, aspergillus and other fungi of veterinary importance, you have to find out, and you have to put this part or this morphology as your assignment. The last slide, that is what other stain we can use beside lactophenol cotton blue. These are enlisted that this stain can be used, but generally this stain are not routinely used in every laboratory. Lactophenol cotton blue is very easy to perform, and that give very good contrast, and that still preserve the structure. That's why generally used in almost all the laboratories. But beside this one, lactofuscin stain and aniline blue stain can be used in place of lactophenol cotton blue, and these two stain or both the stain have similar principle, and that will give similar color to the fungi. So with this, I would like to end today's class. And before ending, I would like to give one assignment, that you have to find out various veterinary important fungi, and you have to draw macroscopic and microscopic morphology of that fungi. Okay, so thank you everyone for listening me and giving me this opportunity for presenting lactophenol cotton blue to you. If you have any query, you can divert me on this one. Thanks everyone.